Hello everyone. Today we want to continue our discussion of protein function by discussing the mechanism of oxygen binding and releasing from hemoglobin in a cooperative way. So the mechanism of oxygen binding cooperativity involves the conversion of hemoglobin from two different forms, R meaning relaxed, which is the oxygenated form, and T for tense, which is the deoxygenated form. So in the conversion from the T to R form, when oxygen binds, one of the alpha-beta dimers rotates 15 degrees with respect to the other to form the oxygenated hemoglobin. So that's shown in this diagram, which maybe isn't all that clear. But in this diagram right here, this animation, we can see that we have the 15-degree rotation. And you can, if you look up here where the red is, that's the oxygen. So you can tell which form is which. So that's oxy, deoxy, oxy, deoxy. So oxygen can bind to the hemoglobin in the R form, and it doesn't bind in the T form. So here's a diagram of the structure of the T form. So I wanted to point out a few things. One is that there's these specific contacts that occur between alpha and beta subunits in the T form. The other thing I wanted to point out was the size of the cavity. So there's a large cavity inside of the hemoglobin when it's in the T form. When that 15-degree rotation occurs, so that we're in the R form, then I'm going to circle these contacts again. So what happens is that the beta chains are brought closer together. So the alpha-1 beta-2 and the alpha-2 beta-1 interfaces, those have changed. I have a close-up I'll show you in a second. The other thing I wanted to point out is I drew the same arrow so it was the same size as the previous slide. The cavity of the R form is a lot smaller than it is in the T form. So let's look at those changes in the interface. So this is specifically the alpha-1 beta-2 interface. So in the T form, which is over here on the left, the histidine makes these specific contacts with these 3 and pro residues on the alpha chain. But when that 15-degree rotation occurs, when you convert from the T form, which is shown here, to the R form, which is shown here, you can see that the histidine moves way up right here, and now it is contacting these two 3 residues. So an intermediate conformation is not allowed, which sort of well, how could that be? How does it move from one to the other? But it's more of like a digital rather than an analog conversion. It's like a digital clock when you move from one number to the next without moving in between, the way a clock with a sweep hand would. So what we should do now is we should zero in on what's happening at the oxygen binding site and see what's happening there. So in that 15-degree rotation that occurs, if the hemoglobin is in the T form, the conformation of the protein in the T form is such that the histidine, remember the histidine is part of this F helix, so the his is the 8th residue in the F helix. Remember there's an actual covalent bond between the nitrogen of the histidine and the iron. And so when the his moves, the iron comes along with it, and the iron is bonded here. There are 4 bonds to nitrogen in the porphyrin ring. So that causes this porphyrin ring to pucker a little bit. It pulls the iron out of the plane of the ring, and it makes it really difficult for oxygen to bind when the iron is in that configuration. So when hemoglobin is in this T or this taut form, it has a low oxygen binding affinity because the iron is pulled out of the plane of the porphyrin ring. When the conversion occurs from the T form to the R form, if you look at the histidine right here, that 15-degree rotation moves that histidine much closer. And now the iron is in the plane, and now this porphyrin ring is nice and planar, and the oxygen has the ability to bind. So we can see this if we go to the models of the protein data bank. So if I click on this link right here, I get to the protein data bank. So if we look at this animation here, we can see that we're going between the deoxy and the oxy form. The oxygen is shown in this kind of cyan or bluish color. So, so we have oxy, deoxy, like that. This red right here, that is the porphyrin ring. This yellow right here, that's the histidine, that's the proximal histidine. the F8 histidine. And this yellow piece right here, that's that whole F helix. So if you look at the whole molecule, if you look at the 15-degree rotation, you can see there's a conformational change that involves that F helix, where the helix either pull in or out like that. So it's either the histidine is either close or far away from the planar porphyrin ring. So if the histidine is farther away, then the iron is pulled out of the plane of the ring. And that's actually not shown here, but in that case, the oxygen can't bind. But when the oxygen when the F helix moves so that the histidine is much closer, then the porphyrin ring is in its planar configuration and the oxygen can bind. Here's another picture right here. So this shows an overlap of the deoxy form, and it's shown in blue. So you can see that the histidine is further away, and this is the rest of the F helix right here. And here we show the porphyrin ring in blue. When the T to the R form, when that 15-degree rotation occurs, and the R form is shown in purple, you can see that the histidine is much closer, and this porphyrin ring is is a planar configuration, and now the oxygen can bind. So here's another animation that shows, I can do this a couple times so that we can see this. So this shows here's the histidine F8, and this whole barrel right here is meant to represent the F helix. The porphyrin ring is shown kind of in this puckered conformation right here. And if we just show the conformation from T to R, like that, the histidine is much closer, the iron is in the plane, we're nice and flat, and the oxygen can bind. So if I go and do this a couple times, and you can see there's the T, and there's the R. So if you kind of flip back and forth, you can see that there's the 15-degree rotation causes this shift in the movement of the F helix. So here we are, deoxy and oxy, like that. Here's another picture. So again, this shows the F helix. Here's the histidine, and it's a little bit further away. The iron is shown. You can see it's not completely centered, and you can see that this porphyrin ring is not completely planar. But when the 15-degree rotation occurs, the histidine is closer to the iron. Now we're nice and centered, and now nice and planar, and the oxygen can bind. So the shift in the position of the F helix when the heme binds to oxygen, that's the adjustment that triggers the T to R transition. So what keeps hemoglobin in this taut or the T conformation? The most important takeaway from from today's talk is that ion pairs stabilize the T form. This is a very highly schematic diagram right here that shows all 4 of the chains of hemoglobin as long lines, which obviously they aren't, but it's drawn this way so that we can see all of the ion pairs. So we can have ion pairs forming between a positive his and a negative asp, between the negative carboxylate and a lysine, between a carboxylate group at the end of one chain to the amino group of the other chain. So all of these are examples of ion pairs that can form, and ion pairs stabilize the T form. So this picture is nice because it shows what one of these what it actually looks like. So here's a histidine right here. So when the histidine is positively charged, it can form an ion pair with this negatively charged asp. The other thing to note is that this histidine happens to be at the carboxyl terminal, so it has a carboxyl terminal right here, which is negative, and that forms an ion pair with a positively charged lysine. So this shows a couple of ion pairs all within the same region of the hemoglobin, but as you can see from this schematic diagram, they occur in several locations throughout the molecule. So here's another picture right here. So this is ion pairs in the alpha chain. Here we have in the beta chain. So in this picture, this shows the deoxyhemoglobin. So ion pairs are shown as dots. What I did is I circled the ion pairs that are disrupted when the hemoglobin transitions from the T form to the R form. So we don't break all of the ion pairs, but a number of them are broken. So how do we go from the T to the R form? What happens is one oxygen has to be able to bind to the T form. And it's difficult for that to happen because remember the iron is pulled out of the plane of the ring. So one oxygen has to manage to do that anyway. And when that happens, that strains the molecule, breaks the ion pairs, and there is a complete conversion where hemoglobin snaps from the T form to the R form. So the conversion happens all at once. So all 4 subunits snap from the T form to the R form. So once you form the R form, the subsequent molecules of oxygen bind more easily, and that's the basis of cooperativity, that it's difficult for the first oxygen to bind, but then it gets easier and easier and easier for subsequent molecules of oxygen to bind. So this is a description of an allosteric protein. Allosteric proteins usually have more than one subunit. There's usually 2 conformations of the allosteric protein. So hemoglobin is allosteric, myoglobin isn't. When oxygen binds and then dissociates from myoglobin, there's no change in the structure of the protein. Another important feature of allosteric proteins is that they have allosteric modulators which influence oxygen binding to hemoglobin. So this does not occur with myoglobin. So with hemoglobin, those allosteric modulators, there are 3 main ones that are CO2, protons, and bisphosphoglycerate. Allosteric means other place. These 3 allosteric modulators don't bind to the oxygen binding site. They bind somewhere else. But when they do bind, they communicate information to the oxygen binding site. So we want to see how that happens. So we're going to consider CO2 and protons together. This describes the Bohr effect. This is after Christian Bohr, who all the way back in 1904, before anybody even had any idea of what proteins looked like, noticed that carbon dioxide and protons could influence hemoglobin's ability to bind oxygen. So this is Christian Bohr. He was actually the father of Niels Bohr, if you have heard of the Bohr atom in general chemistry. So the reason we consider CO2 and protons together is because in the muscle cell we have myoglobin, and myoglobin releases its oxygen so that glucose can be oxidized in the presence of oxygen to yield CO2 and water, and a whole lot of ATP. So when your muscle cells are working hard, they need that ATP. But that means we can consider CO2 to be a waste product. So CO2 diffuses out of the muscle cell and into the blood, and it can enter the red blood cell where it will find carbonic anhydrase, which catalyzes the reaction between CO2 and water to form carbonic acid, and carbonic acid can dissociate to form H plus and bicarbonate ions. So that means that a buildup of CO2 indirectly leads to a buildup of H plus. So CO2 and H plus are waste products of this combustion of oxygen process that occurs in muscle cells. So that means that there's going to be an influence of hemoglobin's ability to bind oxygen, and that's going to be influenced by pH. So if we look at this graph right here, this is pH 7.6, 7.4, and 7.2. So you might notice that as you decrease the pH, that if you look at the P50, that the P50 value increases. So as pH decreases, the P50 value increases. So when the partial pressure of PCO2 increases and then indirectly protons increase too, what that does is it increases the P50. And when P50 goes up, the oxygen binding affinity to hemoglobin goes down. And that's a good thing because we want to make sure that under these circumstances, oxygen is delivered to cells. So we can look at an animation of this as well. So if we look at this curve right here, we can draw our lines right here and we can see that as the pH decreases, the P50 increases. So if we were to look at more basic blood, that means that the hemoglobin would hang on to its oxygen more. But what I like to do is I'd like to focus on what happens when the pH decreases. So if the pH drops, which it will in the vicinity of the muscle cell when it releases CO2, which then reacts to form protons, the pH goes down. This arrow right here represents the increased amount of oxygen that can be delivered to cells when the pH goes down. So how is it that pH can influence oxygen binding affinity? When the pH decreases, if oxygen binding affinity goes down, that means there has to have been a conversion of the R form to the T form. How does that happen? Well, hemoglobin acts as a buffer and binds those excess protons. There are a lot of histidines in hemoglobin that can bind protons. And when the histidine side chain binds protons, it becomes positively charged. And then those positive charges can form ion pairs with other negative side chains in the hemoglobin molecule. Those form ion pairs, and ion pairs stabilize the T form. So a good example right here is if we look at that histidine 146, it's not particularly close to, it's not particularly close to an asp when it's in the R form. And in fact, they're not particularly still either. If you look at electron density maps, they're very blurry, which means that these residues are able to, they kind of flop around. It's like taking a photograph of somebody who's running. The photograph is probably going to be blurry. But when you convert to the T form, these are fixed in place, and the electron density maps show that. And what happens is, in the R form, we don't have a proton. In the T form, we do. And when the proton binds, then we have a positive charge, and now we can form an ion pair. And this shows what it actually looks like. So here's the histidine ring right here with its added proton, and then this shows the ion pair that is formed with this asp 94. If histidine 146 is unprotonated in the R form and protonated in the T form, there must have been a pKr change. And in fact, we can look at this graph right here, and we can see if we look at that histidine at the beta 146, its pKr is about 6.5. This represents the R form right here, and the R form, and it increases to almost 8 in the T form. So an increase in the pKa means there's going to be an increase in proton binding affinity. The pKr of the His 146 is much higher in the T form than it is in the R form because it accepts that proton and then forms the ion pair, and ion pairs stabilize the T form. So if we look what happens in the lungs, hemoglobin that has bound to protons when it was in the tissues, when it comes back up into the lungs, the ion pairs break when the protons dissociate. And protons combine with bicarbonate, which is shown in this equation down here, to form carbonic acid. This dissociates to form CO2 and water. And then the CO2 in water, the CO2 aqueous that's in the blood, passes through the capillaries of the lungs, and the CO2 is exhaled. So that way we get rid of our CO2 waste product, and indirectly we're getting rid of the protons as well. So now that the concentration of protons is not as high now that we're in the lungs, and the oxygen partial pressure is much higher now, hemoglobin makes the transition from the T to the R form, and now it binds oxygen in the lungs and then returns to the cells to deliver that oxygen to the cells. Well, it turns out that carbon dioxide not only influences proton concentration, it also can act as an allosteric regulator in its own right. It can react with the amino termini of the chains in hemoglobin to form something called a carbamate. And this carbamate is negatively charged, and this negatively charged carbamate can form an ion pair with another positively charged side chain, making an ion pair, and ion pairs stabilize the T form. So if we're going to summarize the Bohr effect, in the lungs, hemoglobin arrives. It has bound its protons on all of those histidines. It has formed the carbamates, so it's bound to CO2. it encounters a high concentration of oxygen. So Le Chatelier says we're going to go in this direction. So that means we're going to drop off the protons, drop off the CO2, and pick up the oxygen. So a change goes from the T form to the R form. So you have lots of ion pairs formed right here, and the ion pairs break when we convert from the T to the R form. Now, the hemoglobin with its oxygen travels down to the tissues where it encounters H plus and CO2, which are waste products that the tissues produce. And now we're going to go in that direction. If we have a high concentration of protons and CO2, then the hemoglobin will drop off its oxygen. The histidines bind the protons, the carbamates form, we're in the T form, and now oxygen is delivered to the tissues, which is what we want. So this is summarized in this diagram right here. So CO2 from the cells in the tissues diffuse through the blood and into the red blood cells. Carbonic anhydrase is found in red blood cells to form carbonic acid, and here is our hemoglobin with all four of its oxygens bound. So a proton dissociates from the carbonic acid to give us bicarbonate, and that can diffuse into the plasma. So now here we have hemoglobin with its protons. Ion pairs are forming. Now we've gone, this is the R form, that's the T form. We dump off the oxygen, and the oxygen is delivered to cells of the tissues. In the lungs, the opposite occurs. So here the hemoglobin arrives from the tissues with its protons. And now what happens is a proton can be donated to bicarbonate to form carbonic acid, and oxygen from the air can now bind to hemoglobin. So now we've gone from, this is the T form, now this is the R form. The reaction occurs in the opposite direction. Carbonic anhydrase can hydrolyze the reaction in both directions. So we get H2O and CO2, and we expel the CO2 in the air, and now we have fully loaded hemoglobin with its oxygen. It can go to the tissues and start the cycle all over again. So those are two allosteric effectors, H plus and CO2. There's a third one, is bisphosphoglycerate. So bisphosphoglycerate looks like this. Sometimes you'll see it as diphosphoglycerate in the older literature. It's the same thing. This is more of a chemically correct name. So bis and di both mean the same thing. They both mean two. But so if you have something like ADP, adenosine diphosphate, that means the phosphates are next to each other. They're covalently bonded next to each other. Bis means that they are two phosphates, but they're not bonded to each other. So this is 2,3-bisphosphoglycerate. You can see lots of negative charges. These phosphate groups have two negative charges apiece, and we have a carboxylate group, which is a negative charge. Remember at the beginning I said that the T form had that large central cavity, and it turns out that BPG fits really nicely into that cavity, whereas the cavity of the R form is too small. So we can use Le Chatelier again. Hemoglobin can bind either to BPG or oxygen, but they both can't bind at the same time. So when hemoglobin is bound to BPG, it's in the T form, and that's what's being shown right here. So here's the BPG with those 5 negative charges, and there are positive charges lining this cavity. So we can see we have histidines, we have lysines. Here's another lysine right here. We have the amino terminal. Here's another histidine. There's another histidine. Here's another lysine. There's the amino terminal. So we have the amino terminal, histidine, and lysine residues, all of which are positive, and they form these really nice ion pairs with bisphosphoglycerate. And ion pairs stabilize the T form. So here's another picture right here. So here's the 2,3-bisphosphoglycerate. Again, here's positively charged histidine, positive lysine, positive histidine, positive histidine, positive lysines, and we have this amino terminus right here is positive. That amino terminus is positive. So we have this nicely lined, positively charged cavity, and it's a really nice place for BPG to pop in there and form a whole ton of ion pairs. So if we do an experiment in the lab and we have our hemoglobin with no BPG, we can see that there's a shift to the left. So the P50 goes way down. So if the P50 goes down, the oxygen binding affinity goes up, and that's not good because if hemoglobin is hogging the oxygen for itself, it's not going to be delivered to the cells. So having BPG is really necessary for delivery to cells. So in the absence of PPG, the P50 decreases, that stabilizes the R form. Now we want the R form in the lungs, we don't want it in the tissues, because if we have the R form in the tissues, oxygen won't be effectively delivered to the tissues. So this is interesting to look at fetal hemoglobin. So fetal hemoglobin has an alpha chain, which is the same as the human hemoglobin, but instead of a beta chain, it has a gamma chain. So it's synthesized from a different gene. It's almost the same, except instead of a histidine at 143, it has a serine. So if you look at the histidine right here with its positive charge, if you have this histidine here and this histidine here, and in both cases you've changed it to a serine, you've lost 2 of those positive charges. So that means that hemoglobin F doesn't bind BPG as well. So as we saw in the last slide, not binding to BPG means that you are going to favor the R form. This is good for the fetus because you have to be able to transfer oxygen from maternal to fetal circulation. So interestingly, the gamma chain is synthesized, it's made about 3 months into gestation until 6 months after birth. And then around 6 months after birth, there's a genetic switch from the gamma chain to the beta chain. So I wanted to talk about sickle cell hemoglobin. So you might have heard about this in your Gen Bio classes, but now we have some more structural information for us to understand it on a deeper level, I think. So red blood cells have these nice concave discs. They can squeeze through capillaries. They can tolerate slight distortions as they pass through capillaries. This is a highly schematic diagram, but here this shows the T form. The T form is over here. So this is deoxygenated hemoglobin, and this is the R form. It's very highly schematic. Each of the changes shown as a little box. Well, it turns out that the T form has a nonpolar pocket on its outside. It's not something you'd expect. Usually you expect nonpolar amino acids to be on the inside, but it just happens to be this way. There's this nonpolar pocket on the T form of hemoglobin. If things are normal, there's no consequence to that. The problem is an individual with hemoglobin S or sickle cell hemoglobin, that's a mutation in which instead of a glutamate, which is negatively charged and polar, it's changed to a valine. And valine is nonpolar, as you know. So that forms a sticky patch on the outside of the hemoglobin. So again, if you're in the R form, there's no consequence. But what happens is when the hemoglobin becomes deoxygenated, as it does when it travels through the capillaries, the hemoglobin can polymerize. So I'll show you some pictures in a minute. The polymerization of hemoglobin distorts the shape of the erythrocytes, and they don't pass through capillaries quite as easily as the biconcave discs do. So this is what happens. What happens is, okay, so here are 3 subunits right here. So that the valine right here is shown as this blue patch right here. And what happens is that fits really nicely into the nonpolar pocket of one of its neighbors. So that's the schematic diagram right here. So you have the nonpolar pocket, which is just naturally present on the T form of hemoglobin, and the valine fits into one of its neighbors, and that's why the hemoglobin forms these fibers. So here's another picture right here. So here's the valine, which forms it's able to fit really nicely into this nonpolar pocket. So I have a video here that I can show you.
Sickle cell anemia is a genetic disease that affects hemoglobin, the oxygen transport molecule in the blood. The disease gets its name from the shape of the red blood cells under low oxygen conditions. Some red blood cells become sickle-shaped, and these elongated cells get stuck in small blood vessels, so that parts of the body don't get the oxygen they need. Sickle cell anemia is caused by a single letter change in the DNA. This in turn alters one of the amino acids in the hemoglobin protein. Valine sits in a position where glutamic acid should be. The valine makes the hemoglobin molecules stick together when oxygen tension is low, forming long fibers that distort the shape of the red blood cells, and this brings on an attack.
So the clumping of the sickle cell into the capillaries can be very painful from a person who suffers from sickle cell anemia. So you'd think maybe that the gene for sickle cell anemia might have died out, but it turns out, excuse me, that if you if you're heterozygous, if you have one normal gene and one sickle cell gene, that there you just have a little bit of sickling. It's not as much as somebody who's homozygous for sickle cell. And what happens is there's this complicated life cycle of the plasmodia that causes malaria, and it increases the acidity when it's inside the red blood cell. That favors the T form, as we just talked about. Excuse me. And increases sickling. So sickling damages the red blood cell, so the spleen removes the damaged red blood cell and the parasite. So if you're heterozygous for sickle cell, you are protected against malaria and you don't have sickle cell. So you're in better shape than somebody that either doesn't have sickle cell disease at all or somebody who's normal. So that ends today's discussion of hemoglobin cooperativity. I hope it was useful, and I will see you next time.